Welcome. We're excited that you can join our discussion of de-risking strategies for schizophrenia clinical trials. Our panel includes an all-star cast. Dr. Jeff Lieberman is one of the pioneers in elucidating the pathophysiology and pharmacology of schizophrenia. Among his many contributions, he played a key role in the clinical characterization of clozapine and was a driver and principal author of the clinical antipsychotic trials of intervention effectiveness known as CATIE. Dr. Alan Kott is an internationally recognized authority on the elucidation of patterns in blinded data associated with error variance, placebo response, and fraud. Dr. Kott has been with Signet Health and its predecessors since 2005. Dr. Chris Murphy has decades of experience training raters, evaluating ratings quality, and determining what represents an optimal patient for the clinical trial. Now, we have a very positive background for today's discussion, and that at least 13 atypical antipsychotics of multiple formulations have been FDA approved for schizophrenia. And very notably and happily, Cobenfy, the first of a new class of non-dopaminergic antipsychotics, has received FDA approval. Next slide, please. On the other hand, there are many reasons for concern. Most of you are familiar with this meta-analysis from Dr. Stefan Leut and Dr. John Davis and colleagues that shows on the left-hand panel an increase in placebo response in acute schizophrenia trials over the last several decades. And on the right side of the screen, that drug effect has remained relatively constant. The effects on signal detection are obvious. Not shown on this slide, but also of great concern, is that in clinical trials of stable subjects with schizophrenia with predominant negative symptoms, no drug has proven to clearly be superior to placebo. Let's go to the next slide, please. These are just a few examples of the risks to trial outcome that keep all of us up awake at night. Of course, many of these risks are heavily compounded in the critical phase 2 to 3 transition, where error variance may be amplified from increased numbers of countries, sites, raters, and subjects, and placebo response may markedly increase from expectation bias in the wake of robustly phase 2 results. Then on top of this are the wildcard challenges that have been peculiar to the last several years, such as the impact of the COVID pandemic and the unavailability of familiar high-performing sites in Eastern Europe due to geopolitical conflict there. It is my great pleasure to reintroduce Dr. Jeffrey Lieberman, who will provide us a historical and global context of how we may de-risk these challenges and more. Dr. Lieberman?
Thanks, David, and hello, everybody. Pleasure to be speaking to you on one of my favorite topics, which is psychopharmacology. But at the same time, we have to admit that psychotropic drug development is not for the faint of heart, meaning that the brain is tough and it's more difficult than other organ systems. And I think it's notable that in neurology and in psychiatry, there's been progress, but it's not been as prolific as in other disease areas or other organ systems like oncology, cardiovascular, infectious disease, etc. But it's not because we don't try and we're not as smart. It's because the brain is tough. It's a singularity of organ and in terms of its complexity and multiplicity of function. On the other hand, there's tremendous opportunity, both clinically and commercially, for those who are able to meet the challenge. So we're going to try and outline some of the key issues to bear in mind to optimize the likelihood of success. Next slide, please. So the current landscape is one in which there are many antipsychotic drugs that are currently marketed, and they're pretty much not all the same, but they focus on Positive symptoms is the main pathological target, and they're also targeting the dopamine 2 receptor isoform, or the 2,3 family isoform. And we have limited variety in terms of the novelty of the mechanisms of action, although that is changing in terms of the recent drug approvals. On the same time, people, I think, are aware of the fact that the more you try and develop novel mechanistic of action drugs, the higher the risk and the uncertainty of the gain. It's not to deter people, but it's just to let everyone know what the situation is in terms of many have tried, and only recently we've had some successes with drugs like Karuna's KarXT and then now Cobenfy, and soon to be coming other compounds. When developing the drug, the question is are we trying to develop a comparable drug that has maybe some incremental benefit over the existing compounds that are currently available? Or are we trying to develop something that actually adds incremental superiority in efficacy or safety? It's, from a therapeutic standpoint, the ways to separate are having a drug that is effective in patients whose symptoms are resistant or residually symptomatic to current treatments, like clozapine distinguishes itself as doing, or identifying the other symptom dimensions of schizophrenia as their therapeutic targets, notably negative symptoms, cognitive symptoms, and maybe affective stability as well. Ease of administration shouldn't be underestimated as an important factor also. Anything that can be once a day, anything that can be longer acting is clearly desirable. And in terms of mode of administration, the question is whether the drug that's in development is being developed and studies are designed for it to be used monotherapeutically or adjunctively. Now, almost all of them have been developed as monotherapeutic agents. On the other hand, if you look at your clinical populations, hardly anybody's been treated with only one drug. There's multiple drugs that people are receiving, which tells you about something in terms of clinical practice and the complexity of treating a disease like schizophrenia. And it's not that different from, let's say, cardiovascular disease, where in hypertension, you can be getting a beta blocker, a diuretic, calcium channel blocker, et cetera. And I think the norm is going to be in the future a rational form of polypharmacy, which now de facto is occurring, but is really kind of ad hoc. Another issue in terms of mode of administration is the ease of administration in terms of whether you require dose titration, let's say like clozapine does, or you can start at the therapeutic dose right off the bat. And of course, duration of action. We have long-acting injectable medications. It would be nice to have an oral long-acting medication at some point. Next slide, please. So this is a list in the OS of the FDA-approved medications, and in the dark coloring, the black font is the first generation drugs, which act through a mechanism of action of binding with high affinity to the dopamine 2 receptor, acting as a full antagonist, and the duration of occupancy of the receptor usually exceeds the dosing interval, which means that the pathway is shut down 24/7. This was, of course, sort of improved upon or iterated with the introduction of the second generation medications, beginning with clozapine, which acts as a full D2 receptor antagonist, but binds to the receptor with lower affinity and doesn't occupy the receptor for longer than the dosing interval. And by doing so, it educated us to the fact that you can achieve antipsychotic effects by intermittent interruption of the hypothesized overactivity of dopamine in the mesolimbic system. The other characteristics of the second generation drugs are they have affinities for other neuroreceptors, most notably the 5-HT2A receptor, although the contribution to the therapeutic effects of the second generation drugs besides its D2 affinity has not been established definitively. Aripiprazole, brexpiprazole introduced another iteration on D2 targeted treatments, that of partial agonism. So these drugs bind with high affinity to the dopamine 2 receptor or dopamine 3 receptor, but they have intrinsic activity of stimulating that receptor. Intrinsic activity, which is anywhere from 20 to 40% of what the endogenous ligand has, dopamine. So it's not completely shutting down that pathway in terms of activity, but it's dampening it so it can't go above what the intrinsic activity will permit. Caplyta or lumateperone introduced yet another iteration, a compound that had affinity for the dopamine 2 receptor, but also complex pharmacology at other receptors beyond which is just the 5-HT2A receptor. I'm not going to go into it, but it really was a very different and novel D2 plus compound. And then most recently, we've had the introduction, a milestone in psychopharmacology, which was the development of an antipsychotic drug that was therapeutically effective, but had no affinity for any of the dopamine receptor isoforms. And you could say that pimavanserin exemplified that, which is true, which was purely 5-HT2A for the most part. On the other hand, its efficacy is really very limited at best. But that wasn't the case for xanomeline and trospium or Cobenfy, which has been approved, is the first non-D2 targeted effective antipsychotic drug. But there's issues with that we'll get into in a second. Next slide. So here, these 30-odd drugs, how do they differ clinically? Well, not that much. More in side effects than efficacy. The efficacy, as I mentioned before, is mainly focused on positive symptoms. No matter what claims are being made, I will stake my opinion, my reputation that the APDs, as they are now, do not have genuine therapeutic effects on negative and cognitive symptoms. We can debate that if you want, but that leaves open the opportunity to be developing therapeutic agents which target those other symptom dimensions than positive symptoms. And the reason why that I think is going to be necessary is because the psychopathologic dimensions of schizophrenia, psychosis, negative symptoms, cognitive symptoms, are underpinned by different pathophysiologic mechanisms. At the same time, you can, if you want to, find a hierarchy of efficacy against positive symptoms. Clozapine, of course, is distinguished by virtue of its efficacy in treatment-resistant patients, and followed by that, and this really follows the Cady study's findings, the Eloit and Davis et al. Meta-analyses, the UFES study, etc. Clozapine being the most effective, followed by olanzapine, followed by amisulpride, and then risperidone and lurasidone. There's a cruel irony, though, in the fact that there seems to be a correlation between treatments that are the most therapeutically effective as having the most side effects, with clozapine being the exemplar. Everybody knows what the side effect liabilities are, and olanzapine, even though it doesn't have as onerous ones as clozapine, is still a problem with the fact that it induces weight gains and endocrinopathies. If that relationship is coincidental or is actually built into the mechanistic aspects of the drug's effects, we're not sure, but that's something that we have to contend with. Next slide. So these are some of the drugs that are in development recently and currently. I'm not going to go through all of these other than to say that the target that beyond dopamine that seems to be the most active in terms of development is in the glutamate pathways, one of the glutamate system targets in the synapse, and also the muscarinic pathway, specifically the muscarinic receptor subtypes of M1 and M4. And of course, that's what Cobenfy was based on. There's also a drug which is kind of an old new drug, which is amisulpride, which for reasons that are mostly logistical as opposed to anything that's really justifiable. Amisulpride has not been able to be brought to the United States in the form that it's used in Europe, but LB Pharma has figured out a way to do this by modifying the molecular structure, and I'll show you what that is in a moment. So that's kind of the old new drug. Next slide. So this is really a series of graphs which show you the success rates in the different phases of FDA development by organ system or indication. And you can look at this at your leisure, because I think we'll make the slides available, but all indications, the rates are in phase 1, a drug making it all the way through is 8%. If they get to phase 2, what's the chance of it succeeding going all the way? It's quite about 30%. Now, if you look at psychiatry, though, psychiatry's chances in phase 1 are 7.3%. In phase 2, it's 26%. And in phase 3, it's 55% or 56%. So this is really a gauntlet that drugs have to navigate, and sponsors have to be able to steer them through at great expense. And this is called, for that reason, the valley of death, meaning that there's a lot of risk of failure of drugs not succeeding and getting the approval and generating revenue stream and a return on investment. Hence the need to improve the quality and efficiency of our drug development process. Next slide. So the reasons for drug failure have been nicely alluded to by David in his introduction. They're going to be commented on further, so I'm not going to go through them in any detail other than to comment on a couple of them. So the first question is, if a drug isn't successful, the question is it the target the drug is trying to address, the molecular protein that it has affinity for, or is it the drug that's failed, meaning it hasn't sufficiently engaged the target or activated the mechanism that the target works by, that is the problem. So you can pick a wrong target because you think that that target is integral to the pathophysiology of the illness. But if it's not, then even if the drug binds and acts the target as the way it's supposed to, it's not going to be effective. Similarly, if you have the right target, but your dose is not necessarily engaging it properly or is not achieving sufficient activity, occupancy of the target, it's not going to work. Related to that is dosing. Many mistakes are made in terms of dosing a drug when it goes into clinical testing. Not enough time is spent on mapping the dose response curve in terms of the range that defines its therapeutic activity, its maximum tolerated dose, its minimum effective dose. And as a result, companies can totally miss the signal of a drug that could potentially be very effective. All of the other things, from design to site selection to rating to fidelity of the protocol adherence, they all roll up to the issue of enhancing signal to noise detection, meaning being able to see the signal of if the drug is effective without getting obscured by the noise and rising placebo rates. How you can maximize the success, apart from minimizing the reasons for failure, couple of things to mention. Fewer sites are better than more sites, but if the quality control is good, then you can use more sites. Sample enrichment is a bit another technique. Examples of sample enrichment include, when the cholinesterase inhibitors were developed for Alzheimer's disease, and Aricept being the first, Ken Davis and the group that really pioneered some of that work, they picked their sample based on demented people who showed sensitivity to atropine administration, meaning it impaired their memory. So these individuals were selected among those with Alzheimer's to be selectively responsive to a drug that enhanced muscarinic activity, and it proved to be the case. Another way would be to have a placebo lead-in to a study. So you have a placebo lead-in to try and weed out the placebo responders before randomizing to your active experimental compound and then placebo. Of course, this adds time and expense. Another way would be to isolate the sample. So with clozapine, it was treatment-resistant patients. And another instance, it could be individuals who are early stage in their illness or first episode patients. These are things that can improve the likelihood of success. At the same time, there are also things which diminish the generalization to real-world experience. Next slide. One thing that's really overlooked for the most part is that schizophrenia has a lot of variability and heterogeneity, but even at the etiologic level, which means that it wouldn't necessarily be responsive to the same therapeutic agents. Within idiopathic schizophrenia, you have genetic variants in that most individuals with schizophrenia, validly diagnosed, are polygenic, meaning they have many genes contributing little amounts of variants that are not very penetrant. But then there is some proportion that have rare mutations, which are highly penetrant. And then you have what are called genocopies, meaning syndromes in which there's a genetic alteration, a deletion or a gain in function that includes genes that affect risk of schizophrenia. And the best known example of that is the George syndrome or velocardiofacial syndrome, where there's a deletion in one of the chromosomes of a segment of DNA in the 22nd chromosome in the Q11 region. And these individuals, apart from their other phenotypic features, have a 30+% likelihood of developing schizophrenia when they enter the age of risk. There are also phenocopies, meaning that there are environmental factors which induce the syndrome of schizophrenia, chronic methamphetamine or phencyclidine abuse. There's something called autoimmune or limbic encephalitis, where an antibody has affinity for a CNS protein. In the case that it was characterized by Joseph Dalmo, it's the NMDA receptor, and some forms of temporal lobe epilepsy. And finally, individuals can present with symptoms of schizophrenia, which are what are called facsimiles, meaning there's no biologic basis that it can be attributed to, but they're related to the psychological phenomena of dissociation. Next slide. Okay, so here's some examples of drugs that have either succeeded or failed for reasons that potentially could have been avoided. This is the TAR1 agonist, the Synovian developed ulotaront. It's not dead. Other companies, in this case, Otsuka, has picked it up, but it had positive phase 2, and then it failed to separate in phase 3. That's not an uncommon occurrence, an expensive one, and one that could be avoided, presumably. The question is why? And in this case, we don't know if it was the target was wrong, or it was the dose that was administered, or other factors which undermine the signal-to-noise detection in the phase 3 drug protocol implementation. Next slide. Now, glutamate, as I mentioned, after dopamine, is really the most actively pursued target in addition to the muscarinic pathways. And the glutamate synapse, for good reason, is a very complex synapse with axon and a dendrite, with an interneuron, and it has multiple potential targets to modulate this pathway. You can do it postsynaptically by allosteric modulation at the NMDA receptor. You can do it presynaptically by inhibiting the release of glutamate, or you can alter glutamate's uptake into the glial cell supporting cell and its metabolism to be recycled into glutamate again. The one target that's been pursued most actively is really the allosteric modulation of the NMDA receptor. And if we go to the next slide, the first example that really made a strong effort in this area was Roche's bitopertin, which developed a compound that didn't just act at that modulatory receptor site in postsynaptically, but it inhibited the uptake of glutamate, which was the amino acid that acted as an allosteric modulator. Bitopertin was successful in phase 2, but as Alan's going to elaborate on, in phase 3, it did fail for reasons that are still being pursued. Next slide. Another glutamate-targeted treatment was pomeglumitad, which acts by targeting presynaptic receptors, the 2,3 metabotropic receptor that gates the release of glutamate. And this is an interesting example again. So on the right panel is the phase 2 study published in Nature Medicine in 2010. And the results here were just stunning. I mean, too good to be true in the sense, the white shows you placebo, patients worsened who were on that. The red shows you, I'm sorry, the Purple shows you the active comparator, which was olanzapine. They used olanzapine because this was a Lilly compound. And the experimental drug pomeglometad is in red, which shows you it had a parallel curve or trajectory slope to olanzapine. That's, as Larry David would say, pretty good. And sailed into phase 3. 5 studies later, failed to separate. What's that all about? Well, it's any one of a number of reasons, but turns out that the most likely reason, in my opinion, was they used the wrong dose. The highest dose they used was 80 milligrams a day, 40 BID. Next slide. And Lilly itself did a study which we replicated with an NIMH grant, which showed that in order to achieve an effect of inhibiting glutamate at the glutamate synapse from stimulating the NMDA receptor, you needed to use a dose that was at least twice the highest dose they used, 160 milligrams. Now, this is data which I'm not going to take the time to go through, but it basically shows that the dose that inhibited hemodynamic response in healthy controls to ketamine administration was 160 milligrams of pomeglometad, not 80. 80 was not effective. So the dose that would be needed would be at least 160 or higher in terms of achieving a therapeutic response in schizophrenia. Next slide. These are some studies, references for the path of xanomeline to KarXT to Cobenfy. And the point to be made here is xanomeline is a drug that was originally synthesized in the 1980s in Denmark, and then came to Lilly, and then was studied in Alzheimer's disease, and was found to be effective in psychosis as opposed to cognition. But because of side effect reasons, it sat idle for 30 years before Andrew Miller, with the assistance of Steve Paul, no longer then at Lilly, decided to combine it in a Rube Goldberg-esque fashion with trospium, a peripheral anticholinergic inhibitor, and $14 billion later, BMS is marketing it as Cobenfy. So that's to say that the process of moving things along is not as efficient as could be for a whole variety of reasons. And here I think we get into an issue that often occurs in pharma, which is the suits versus the scientists, making business decisions versus scientific pursuit of potentially effective agents. Next slide. So amisulpride has been available. It's originally developed in France for no good reasons. It didn't come to the United States. And the patent expired, so the incentive of doing so in terms of the IP and preferred pricing was no longer feasible. But the principles behind LB Pharma, and it's an interesting story, which I won't go into, but I was fascinated by it when I did hear it, found a way to overcome that by methylating the amisulpride molecule. And the arrow on the panel on the right shows you where the methyl group was attached to the amisulpride compound. Now, methylation in this case wasn't just simply a kind of ornament that was hung onto this molecular structure for the purposes of IP. It actually affects the pharmacologic properties of the compound, and to may extent also alter the clinical effects of this form of amisulpride. We'll find out as this wends its way through development. Next slide. So just go back one second. So the big thing that is the reason why methylation could affect it is that it alters blood-brain barrier permeability. And the pharmacokinetics of amisulpride are such that it has very low permeability, meaning that you give a higher dose to get a small amount into the brain, which is presumably why it produces little EPS, even though it's D2 selective. D2, D3 selective is a full antagonist, but therapeutic antipsychotic effects. And as a result of its high concentration in the periphery, it drives prolactin up since the pituitary sits outside of the blood-brain barrier, but it doesn't drive it up so high because prolactin is pretty much a silent side effect, except for the problems it causes in women in menstrual cycle functioning and fertility. But LB Pharma just successfully did an IPO, raised the necessary money to do phase 3. And so that'll commence imminently. And we should have a new drug within at least within 2 years. And more question will be finding out what effect the methylation on the biochemical structure has on its clinical properties. Next slide. Oh, we're already here. So no, go back to the diagram. So what I wanted to point out here was the fact that there's opportunity to innovate without taking huge risk, meaning that the unmet clinical needs of people with schizophrenia are not simply replicating suppression of psychosis and preventing relapse, but other things that can be identified as therapeutic targets, such as preventing progression, imitating clozapine and being able to suppress refractory or residual symptoms in patients, and also really the untapped areas of negative symptoms and cognitive symptoms, not to mention the fact that the side effect-free drug wouldn't be a bad either. So how you do this will depend on what stage of the illness you want to select your sample from, but this is really unmined territory that I think there's great opportunity for. Next slide. Last thing I just want to mention is that, okay, you've got a drug, you navigate the gauntlet of FDA successfully, you get approval. Your post-approval determinant of success can also be considered in the development process, and these are sort of listed here, but it's important to do so because when a drug is approved by the FDA and commercially available, because of the fact that the clinical trials were designed to gain approval, were very strict, scientifically rigorous, not necessarily generalizable to the broader population. Clinicians aren't sure how to use it. And we're seeing that now with Cobenfy. So this kind of forethought is important to keep in mind. But all in all, I would say there's great opportunity, albeit with a given degree of risk. The whole point of this webinar today in this educational program is to help you understand what are the key things that can de-risk your efforts to develop drugs, which, as I mentioned at the outset, I encourage the individuals in the pharma, biotech, academic community to continue to do, because there's great need and there's great financial and commercial potential. So thanks for listening, and I'll be happy to answer questions if we have time.
Thank you, Dr. Lieberman. Thanks for an excellent presentation and for sharing your perspective on this very important topic. Now, building on that foundation, I'd like to narrow our focus to a more specific challenge, and that is the rising placebo response, and in particular, the impact that data concerns have on this phenomenon. In my presentation, I'll take a high-level look at the issue and the ways how data concerns impact on placebo response and signal detection. I'll actually use 2 specific examples to illustrate the points. And then I'll pass it over to my colleague, Dr. Murphy, who will share concrete specific examples and practical strategies for overcoming these challenges. Data concerns are frequent in clinical trials, and in schizophrenia specifically, we see roughly 27% of data affected by clinically meaningful data quality concerns. As is illustrated on this particular slide, you can see. This represents 17 acute schizophrenia trials. Each bar is a single trial, and the height of the bar is reflecting the amount of data concerns identified in that specific trial. As you can appreciate, not only that the percentage of affected visits is fairly high, but we do see a fairly large variability in terms of the presence of data concerns in individual clinical trials. Now, when I speak about data concerns, what I have in mind are clinically meaningful issues that have the potential to impact on signal detection, placebo response, drug response, and the drug-placebo difference. On the next slide, there are just 4 examples of issues and drawing from a family of many more. I will, in my subsequent presentation, focus on the first 2, the lack of diagnostic confidence. And with that, I don't mean necessarily just a misdiagnosis of the individual subjects entering the trials, but as well issues, say, in acute schizophrenia with the acuity of the symptoms. And my second focus will be aberrant variability, and they're mainly erratic ratings. But otherwise, we do see other issues as well. Baseline inflation, artificially inflated scores at the time of entry into the trial, between and within scale discordances, or unfortunately, cases of pure data fabrication, which is relatively rare, but unfortunately still present in clinical trials. Now, data concerns, and if we move to the next slide, please, as I said, are relatively frequent, but one of the attributes of many of those is that they are non-randomly distributed in the clinical trial data. What you see on this particular slide is one acute schizophrenia trial where there were 58 individual research sites participating in the study. The height of the bars represents the number of patients that were randomized at those sites. And the blue line is actually the percentage of visits that were affected by extreme data variability. In this instance, it were erratic ratings. And what you can appreciate is that for the majority of the sites, no erratic ratings were recorded whatsoever. And there were only a few sites where this was happening, typically with a single or 2 cases at most. However, site 55, the one that is highlighted here in orange, is a site that was a clear outlier. Unfortunately, this site was as well one of the highest recruiters in the study, and therefore the impact of this particular site on the signal is much larger than the impact of the smaller sites that may suffer from erratic ratings as well, maybe even with a higher percentage. So as you can appreciate, the issues cluster and identification of the sources, the sites that are potentially responsible for these issues is paramount to improving the quality of the data. Now, what the impacts are can be seen on the next few slides. The first example is coming from trials that were conducted around 2015-2016, again in acute schizophrenia. And here we looked for high variability and the impact it has on signal detection. On the left side, you can appreciate the impact on placebo response. In blue is the placebo response in those sites that were affected by this extreme data variability. And as you can see, there was almost 43-point improvement on average in these sites. On the other hand, the non-outlying sites had the expected, say, somewhere 15-point improvement in the data. The interesting thing, and not surprising though, is that when we looked at the separation, the sites that were affected by these issues in data variance, at those particular sites, placebo did actually outperform the drug compared to the orange bars that show a clear separation of the drug from placebo in the sites that were not affected. Now, you could argue that this is a single finding. However, we have looked into other trials and we have found similar signals there as well. The next one comes from the bitopertin trials in negative symptoms. And here we have again two panels. On the left, we have the subject level data. So this is looking into the subjects that were affected by erratic ratings in the negative factor of the PANSS. On the right, we then have the same information, but for sites that were affected by the erratic ratings. As you can appreciate, in both instances, the subjects and the sites that were affected by the presence of erratic ratings had significantly increased response to placebo, as well, though, to the actual treatment. And in the sensitivity, post hoc sensitivity analysis, removing both either the subjects or the sites resulted in this particular trial to separate 10 milligrams of bitopertin from placebo. Unfortunately, this was a post hoc analysis, and bitopertin has not moved forward, at least not within Russia in this indication. The next slide then looks at the data from the phase 2 Karuna trials with KarXT. And here, looking into 5 different variability indicators or concerns of potential variability issues, we can appreciate that for most of them, patients who were affected by the presence of these issues would not separate KarXT from placebo, unlike the patients who were not affected by these data concerns, be it erratic changes, large post-baseline change, or the opposite of those, identical or nearly identical ratings and very low variability over time. The only case where actually the impacted group separated more strongly than the non-affected group is the high mean square successive difference group, which represents typically a very large and sustained change over the course, or linearly improving over time. Yet even there, the placebo response was huge, but the response to the treatment was even larger and led to the change that we see here and the difference between the drug and placebo. So these are some of the examples from the family of data variants and their impact on data quality, on signal detection. The next example is looking into the, let's say, eligibility of the subjects. If we move to the next slide, please. This looks again into the Karuna phase 2 trial, and it compares patients who present at baseline with anxiety symptoms in contrast to patients that did not suffer from anxiety at the time of randomization. Now, in acute exacerbation, anxiety is a symptom that we would expect to see. So the absence of that may question the actual acute exacerbation of those subjects. Now, what you can appreciate here is that the patients who had anxiety present strongly separated KarXT from placebo. That's the left panel. On the other hand, patients who did not have anxiety present, here actually placebo outperformed the drug, but the difference was not statistically significant. Since Karuna did run their phase 3 trial, we had the opportunity to look into this same issue in the phase 3 data. And on the next slide, you'll see how that played. Again, on the left, we see subjects that had anxiety present, and there was a strong separation between the drug and placebo. On the right, we then see a slightly different picture than we have seen in phase 2, and that is that both the drug response and the placebo response diminished. However, the reduction in both arms ultimately ended up with KarXT not being able to separate in those patients who did not have anxiety present at baseline. So you can see the impact of the presence of the absence of anxiety, if you will, on the signal. Now, anxiety is an interesting marker potentially of acuity. And I want to show you two more things related to this specific symptom or set of symptoms. The first one is that when we compare clinical trials, and if we go to the next slide, please, if we compare the first 25% randomized subjects into the study with the last 25% of subjects randomized into the study, we'll see that anxiety actually decreases over time. So patients who were entered early had higher levels of anxiety compared to patients who entered late into the same trial. What this may indicate is that the selection of the subjects over time, or the quality of the subjects for the trial, decreases over the course of the trial, and this may have ultimately a potential impact on the outcomes of the trial. And my second example related to anxiety is on the next slide. And it is a sad one because it shows that clinical trials are influenced by external circumstances. What you can see here are the trajectories or the severities, if you will, of anxiety and depressive symptoms as measured by the PANSS in Ukraine compared to the US. US in red and Ukraine in blue. So you can see they basically were stable for most of the time with some variation, but not a large one until 2022, where we can see a very sharp increase in both anxiety and depression in the data coming from Ukraine. And it's obvious what the cause of this increase is. Finally, to conclude my presentation, if we go to the next slide, many of the data concerns that we identify, and many of those that we spoke about today or that I didn't address, such as discordances within the PANSS or across scales, between scales, are present in the screening phase, can be identified in the screening phase. And many of these issues actually increase the probability, or the presence in the screening phase increases the probability that the same issues will be identified after randomization. Therefore, the screening phase offers a unique opportunity to intervene, to retrain if the issues are driven by inappropriate, say, understanding of the scale administration, or to minimize the impact of data coming from sites where these issues are consistently present. We have machine learning algorithms now in place that help us predict the potential of developing or recording data quality concerns after randomization based on the screening data, so that interventions can be taken quickly and the quality of the data improved when it actually matters. So to conclude, data concerns are relatively frequent in schizophrenia clinical trials. As I said, there are almost 30% of all the data are affected. Many of these data concerns are clinically meaningful, have the potential to impact on drug placebo separation, are not randomly distributed, which allows effective interventions. And it's obvious that these data concerns will be most painful in those situations where the power of the trial are relatively low. With that, I will pass it over to my colleague, Dr. Murphy, to give us more specific examples. Thank you.
Thanks, Dr. Koch, for outlining some of the challenges that we face. Next slide, please. So 2 keys to success in overcoming the challenges that were just outlined are careful subject screening and eligibility reviews, and also ensuring rating reliability. These approaches can reduce several risks, including the risk of large placebo response that has undermined several recent programs. Next slide. Screening out inappropriate subjects or subjects at risk for placebo response generally falls under the category here of predict and restrict, where we have the opportunity to weed out subjects who might put the study at further risk. The subjects that we would want to exclude might be those with inflated screening scores or atypical presentations that run counter to protocol expectations, or like Alan mentioned, those with low anxiety who may not in fact be experiencing a true acute exacerbation. We had a case in the past where we were asked to do an ad hoc review of an atypical subject, one who had elevated PANSS scores, but raised some red flags to the sponsor when they discovered that he self-presented to the site asking to be admitted under really unusual circumstances. When we reviewed the audio, the subject was really jovial and affable, really not a care in the world, and it was counter to the severity that was represented on the PANSS. It turns out on further inquiry that the subject was really inflating and exaggerating symptoms, and he reported that he wanted a change of scenery and to switch things up, and he was ultimately screen failed. Subjects undergoing an acute exacerbation are not always the best historians, but careful attention to a subject history, the course and presentation of the current episode, and the subject's motivation can go a long way to excluding these inappropriate subjects that may be at risk for a placebo response. The next image, please. There are interventional strategies, though, that we can use in addition to screening out potential subjects. Site and subject awareness of factors that contribute to the placebo response are really critical components to any placebo response mitigation. Expectation bias is the main contributor to the true placebo response, and the sites can learn to manage their own expectations and the way they communicate the expectations to the participants throughout the trial. Expectation bias is particularly salient when moving from a successful phase 2 program to a larger phase 3 a program where the enthusiasm for the new compound might intrude upon recruitment and assessment activities, encouraging participants and staff to really help demonstrate the success of an exciting new compound rather than approaching from a more neutral stance where the trial is not treatment and where participants and staff are asked to value improvement, stability, and worsening of the same. Site-facing training workshops and role-playing are effective tools, along with participant-facing scripts and training videos to remind all parties of their respective roles and the conduct of the trial. Subjects and raters can be biased in reporting severe symptoms at screening and baseline and change their approach once enrolled, and this training can go a long way to reducing those risks. Next slide. Site-independent eligibility review can go a long way to ensure that only appropriate subjects are included. This, as I had mentioned before, can include audio review of the diagnostic instrument, the SCID or the MINI, along with scoring of the PANSS to affirm that the site scores are supported by the material. We find that sites are on their best behavior when they know they're being monitored, and early monitoring can help support sites and raters who may not understand the protocol or scale requirements and may not be scoring objectively. We find really a low percentage of subjects who have submitted subthreshold symptoms when we do a comprehensive eligibility review, but that percentage increases when we are not involved in that assessment and conduct retrospective review and find that a larger proportion of subjects have sub-threshold scores on our review at screening and baseline. The risk, of course, is including subjects with inflated entry scores, which can contribute to pseudo-placebo response. Next slide. So as an example, we do suggest a robust documentation of the illness course and key clinical features with site-independent holistic clinical review prior to enrollment. On the left, we can see a typical study may ask for a simple checklist looking for the establishment of an acute exacerbation beginning with a certain time period. A more robust assessment would include documentation of the factors that contribute and are expected in acute exacerbation. And this would include the areas of functioning that have been impacted, clear documentation of worsening from a baseline, that there are subjectively distressing symptoms, again, to ensure that the appropriate subjects are being enrolled. Next slide. Rater training has a challenge of establishing and maintaining valid and reliable symptom ratings within sites and subjects and across sites and regions. The study teams are faced with calibrating new and possibly naive raters, particularly when protocols call for uncommon rating scales such as the NSA 16. There are examples of experienced raters who bring to this study their own idiosyncratic rating rules, and naive raters who have insufficient probing or scoring, and comprehensive training can reduce these risks. Next slide. Would recommend a comprehensive interview skills training in addition to training on scoring conventions. This includes restricting poor performing raters and putting raters on hold for close monitoring, and ongoing performance monitoring and targeting refresher can be very successful. Next slide. Our training does increase inter-rater reliability and decreases the noise in the study. Next slide. Endpoint monitoring is essential to maintaining the fidelity to the administration and scoring guidelines established. And this monitoring can include monitoring for patterns of data quality flags during the scale collection itself, as well as review of the interview and score quality throughout the trial. It can be targeted remediation to the rater site and at study levels, and quality can be tracked over time. Next slide. We do find that after our review and remediation and support of sites, that the difference between the rater and reviewer scores, the discordance, declines over time as sites and raters become aligned with the scoring rules that have been established. Next slide. An analytics program can bring together the quality findings and monitoring throughout the trial. That clinically driven algorithms can identify early sites and raters that have a potential risk to the study, the interpretation of these leads to a timely targeted interventions and follow-up in collaboration with the sponsor and study teams. Next slide. We do find that this shows examples of different sites and the percentage of flagged flagged visits for scale administration and scoring concerns. Those on the left in red with a high proportion of concerns could be targeted for further remediation or held for further recruitment, while emphasis can be on the sites in green that have fewer concerns. Next slide. So a proactive identification of the problematic sites and data can improve the overall quality of the data and increase the validity. The application of the analytics customized with the mitigation strategies can decrease the presence of data quality concerns. Thank you. And I will turn it over to questions.
Thank you, Dr. Murphy. We have a question for you. And the question is, does it matter whether your raters have an MD, DO, PhD, master's, or bachelor's? Is there any evidence on this when controlling for years of experience?
Thanks. That's a great question. In the that we've reviewed, we don't find a difference in the educational background in the ratings quality. We find excellent performance really at all levels of expertise, and unfortunately, we find poor performance as well at all ends of the spectrum as well. The critical factor here is the performance on the training and assessment for both interview skills and training, and that's independent of the degree background.
Thank you, Dr. Murphy. And thank you to the panel for superb and informative and engaging presentations. I wish we had more time for questions, but on behalf of Signet Health and the panel in particular, thank you for joining us, and we welcome your questions offline, and we would be absolutely delighted to follow up with you. Thank you, and have a wonderful